Despite having an identical chemical composition, the genetic material found in a bacterial cell differs considerably from the genetic material of eukaryotic cells. These differences inhibit the production of recombinant proteins in bacterial cells. In contrast to bacterial cells, whose DNA is located in the cytoplasm, the DNA of eukaryotic organisms is found in the cell nucleus. This makes the isolation, taking and cleaning of DNA that is to be used in cloning more difficult. Moreover, the DNA in eukaryotic cells forms highly organized structures, the chromosomes, in which the DNA is bonded with histones, proteins forming structural elements of chromosomes. These prevent the synthesis of RNA by blocking access of the appropriate enzymes to the DNA molecule. As bacteria do not have proteins to enable the separation of DNA from histones, the introduction of a chromosome segment into a bacterial cell will not cause the synthesis of the recombinant protein. Further analysis of the structure of genes in bacterial and eukaryotic cells shows that, in contrast to bacteria, genes found in eukaryotic cells have a discontinuous structure. This means that the segments that code proteins, exons, are separated from each other by sequences that do not code proteins, introns. In the eukaryotic cell, introns are cut off in the process of mRNA processing. Hence, the template DNA that is produced contains only the sequences necessary to produce proteins. If a DNA segment with an intron is introduced into a bacterial cell, the protein obtained will have a totally different sequence than expected.